introducing our speaker, Dr. Maria Luisa Enriquez. Dr. Enriquez is one of the very few scientists in the Philippines who has got the expertise and skills to use cytogenetics and molecular genetic methods in disease diagnosis, particularly various cancers like colorectal cancer, breast cancer, lung cancer, and leukemia. Dr. Enriquez's glorious career spans more than 4 decades, with her active involvement in academics, research, and administrative roles, all done with integrity, professionalism, dedication, and brilliance. She did her postgraduate studies in cytogenetics and fellowship in molecular cytogenetics from Tokyo Medical and Dental University, Japan. She is currently the chair of the Research Utilization Committee of the Metro Manila Health Research and Development Consortium, as well as holding the position of scientist in residence at Salle University, besides teaching and guiding students at UP Diliman and SLMC College of Medicine. She's also supervising the Molecular Cytogenetics Laboratory of St. Luke's Medical Center, besides heading the Translational Research Unit of the Human Cancer Biobank of the St. Luke's Hospital, the first cancer biobank established in the Philippines. She is truly an inspirational figure with an envious energy levels. Today's topic is current approaches for assessing the cytotoxicity and genotoxicity of natural products. Over to Dr. Enriquez.
I'm going to, the topic now assigned to me is the current approaches for assessing cytotoxic and genotoxicity of natural products. So my talk is on 3 parts. First, I will describe how we do natural products research in the LaSalle. And the second part would be on 3D cell culture. And the last part is on imaging. So natural product research at DLSU is actually a collaborative effort among the departments, especially of the College of Science. And it involves the Department of Biology, Chemistry, and Molecular Science Unit Lab of CENSER. So this is a team of dedicated researchers involving students, faculty, both graduate and undergraduate levels. So the research team has produced numerous research publications in ISI and Scopus journals. So Cytotoxicity is simply defined from many sources. It's a method to measure dead cells. But in some cases, it is also a measure of some cellular or intercellular structure, loss of some cellular or intercellular structure functions. So cytotoxicity assay may involve qualitative methods as well as quantitative methods. At De La Salle, we do our NATPROD research basically on cell lines. There is less ethical issues and it's easier to handle. So these are some of the cell lines that we use, and these are all available at the American Tissue Culture Collection. So cell line for colorectal, for breast cancer, for leukemia, whether it's chronic or it is acute. So we used to test, up to now, we test the cytotoxicity property or genotoxicity property of natural products. And natural products are any organic compound synthesized by living organisms, either by primary or secondary metabolic processes. And we can classify them based on their biological function, like whether it's antimicrobial, it is anti-apoptotic, or it's anti-proliferative. And usually we use, the materials that we use come from vegetables, fruits, mushrooms, fungi, and other microorganisms, and of course other materials. But the substances that we actually study in cytotoxicity assays are the secondary metabolites, not the primary metabolites. So these are compounds that are not needed by the plant to survive, but they are produced by the plant for other functions, like social signaling molecules, or for competitive weapons to protect them from predators. And of course, plants that have these properties have an evolutionary advantage. Many, of course, of these functions are still unknown. So from the many publications that the group had, we usually focus on NATPRODs from vegetables, which could be glucosinolates, phenolics, or alkaloids. So to be able to study the cytotoxicity property of these extracts, we need to use markers. And in cytotoxicity assays, usually when the extract or the drug, for that matter, is treated to the cells, the first result is the effect on the membrane integrity. And usually the membrane is destroyed, and this will lead to the leaking out of substances inside the cytoplasm into the surrounding area. And in some cases, you can use dyes that are not permeable to viable cells, but now can enter dead cells. So this is just a table, and I think everybody knows this. We usually use only Presto blue or Alomar blue, a resazurin-based dye. So this is usually the flow. There is an extract that you want to study, a chemical profiling, and then there is the identification of the phytochemicals in the NATPROD, the natural product, whether it is crude or semi-crude, and if it is how do you call this? If it is really a particular product, usually they can be analyzed using NMR. Then we go to cytotoxicity assay, and of course we do the usual procedure of determining the IC50, so forth and so on. And then the genotoxicity assay. And then in some cases we do molecular real-time PCR, the quantitative real-time PCR, to complete the whole cycle. So from the chemical profiling to the cellular basis to the molecular basis. So I will not go into the detail, but these are what we use in the lab. And of course, there is the microplate reader that can be used for the absorbance or the fluorescence measurement. So maybe you will ask, why are we using Presto Blue and not MTT? So the recent articles will say that Presto Blue or the resazurin-based dye are more sensitive than MTT. So as I said earlier, we also used real-time PCR to monitor the expression of genes like the CFOS and the CJUN. Usually this is done prior to treatment of the secondary metabolite and after the treatment of the secondary metabolite to check whether there is a change in the expression of these genes. So the genotoxicity assay that we are using most of the time is the COMET assay. So this is a single cell gel electrophoresis, and it's a very popular assay now being used. And because it's easy, there is a software assistance, and it's very visual. You can see the cells, single cells under the microscope. So the assay can detect DNA damage at the level of single cells. So these are the basic steps of COMET assay, but I don't have time to discuss that. But very important here is the pH in the experiment to be able to measure the breakages in the DNA. So the parameters used are comet tail length, the percentage DNA in comet tail, and the tail moment. So maybe the next figure will tell you that in normal cells, so of course this is already treated with a dye. Usually we use cyber green or any fluorescence dye that can that can color the nucleic acids. So you have the normal cells. If the cell is normal, usually the image is rounded because the DNA has not been fragmented. If the DNA is damaged, of course we attribute it to the presence of the natural product extract. This is the tail that are formed. So when the tail is longer, it means the damage is more or it's more extreme. So this image, you can see a single cells under the microscope. So you can count it and you will see how many are rounded and how many have tails. So there's a software for this available. There are free softwares for COMET assay. So this is the head of the image. So and then of course as the DNA is migrating, the tail is going to be formed. So these images have tails. These one are mostly normal cells. So this is a paper that we published. We used the extract from radish, so Raphanus sativus, and these are different cell lines treated with the extract. And the extract was most damaging to MCF7 breast cancer cell line. And this was published and in fact won a first place in the research poster competition at the national level. So I'm happy that the time that we introduced the COMET assay, people got noticed and appreciated the visual images and easier to understand cytotoxicity parameters. So there are two other papers that we were able to publish. And as early, we introduced the COMET assay actually at the Salle in 2009. And up to now, there's another paper that also won another award in an oral research competition at the national level. And this time we use alugbate. I don't know if you are familiar with the green leafy vegetable. We call that Basilia alba. So this is another assay, which is actually my field, but I'm not going to discuss it. What we do in the lab is to induce breakages with an extract. And if this is a normal chromosome, you will see it. The morphology is intact. But if your extract, like a drug, can induce strand breaks or double strand breaks, the chromosomes will look like this. So there will be breaks, there will be gaps into your chromosomes, and you also can count this. So the second part is the new approach in cell culture, which is a 3D cell culture. Nowadays, we routinely do 2D cell culture. And of course, we have a limitation in the 2-dimension given by your flask and your petri dishes. And researchers have claimed that this kind of conditions is actually not very near to the in vivo conditions of cells in the real world. So it is an unreliable predictors of in vivo drug efficacy and toxicity. So if you're doing natural products, there are very many limitations because we are doing the 2D cell culture. In 2018, with the help of Corning, we were able to organize an organoid cell culture seminar as a post-conference activity of the Philippine Society for Cell Biology. We had limited participants then, but we were introduced to the 3D cell culture. And many researchers and papers now claim that the 3D cell culture is truer to the real environment of the cells, because you will allow the cells to grow and interact with their environments in 3 dimensions. So the cells can grow in all directions, and if you treat them with a natural product or any drug, you will see the response very differently when it is in the 2D cell culture. So the method allows a very good, provides a very good environment for the growth and proliferation of cells. So the 3D cell culture is either a scaffold-based 3D cell culture or a scaffold-free 3D cell culture. So in the scaffold-based, it's either the materials, this is according to the materials, they are hydrogel-based or polymer-based. So in either way, the technology provides the cell a place to grow in 3 dimensions. So these are the gels, which are contains a lot of water, and it's a network of polymers, so they can be made up of natural or synthetic materials. And the polymers are hard base support. Nevertheless, they can facilitate the exchange of oxygen and nutrients and in the cell culture. So this is the images that are provided. Got this from the internet, examples of scaffold structure, and in these spaces here will lie your cells. So the cells will be able to have a 3-dimensional space for them to grow. There are scaffold-free 3D cultures, and in this case, there are no scaffolds, and the cells are made to float in your medium. So in this non-scaffold 3D cell culture, there are different types, but I don't have time also to discuss each type. But the most important structure that is formed in their non-scaffold 3D cell culture is the formation of spheroids. So these are clusters or clumps of cells that are formed, but they are non-adherent, so therefore they can move, and they are allowed to grow into the 3 dimensions. So these are examples of spheroids under the scanning electron microscope, and this is what we see, very different from what we see in your petri dishes and in your flask. So these spheroids will allow cell-to-cell interaction as well as cell matrix interactions. And these are the 2 parameters that are being considered in 3D cell culture. So the cell-to-cell interaction, your cells are allowed to communicate with each other in response to the microenvironment, especially if they are subjected to stress or to chemical extracts that are being added in the culture. So the cell and matrix interaction, of course, is also responsible for generating signals so that the cells communicate, and could be responsible for the cell to proliferate, or to stop proliferating, or to differentiate, or to migrate, or to initiate apoptosis. So of course, all of this involves many proteins. So this is just a schematic diagram of how cells communicate in the culture. So this is a table that summarizes the key characteristics of 2D culture and 3D culture. And these are the parameters. Of course, I will not be able to describe all of them. But as I mentioned earlier, my main idea is that the 3D culture give the cells a 3-dimensional space to grow. So another new approach, this is the last part of my talk, is the live cell imaging. So this is a very advanced technology to study the living cells using a time-lapse microscopy. So in this live cell imaging, you can focus on single cells or in clusters of cells. And you can observe dynamic processes, even up to the molecular level. So it is designed to provide spatiotemporal images and subcellular events. So the advantage of live cell imaging is that you can study cell kinetics. You can trace single cell. If you if you're looking at a population of cells, and if you want to focus on one cell, you can easily do that by certain manipulations in the microscope and in the software. So you can study the cell in a very dynamic condition or environment. So it is a very interesting and exciting platform to analyze the effects of natural products and, of course, the drugs. So in the live cell imaging, the microscope can be a confocal microscope. So the confocal microscope is able to scan the whole image by using a pinhole one by one, and then it will synthesize the whole image until you get the whole image. So if this is the image, what it will do is to capture the image in small areas and then synthesize the image to give you the entire image of the cell. It can also do optical sectioning, so it can cut through the layers of your sample, and then eventually it will do a composite image. So that is confocal scanning microscope. Unfortunately, Salle bought a confocal microscope. We have the microscope already. It's the brand is Leica, and unfortunately, because of COVID, we weren't able to set it up. So we'll be waiting for COVID to die down to set the lab up. And this is not the Leica model, but the confocal, when set up, will have its own incubator. It will have its own supply of carbon dioxide, controlled humidity. So this is capable of doing experiments for long observation, maybe a week, 3 days, a week, or several weeks of experiments. For as long as the cells, you mimic the natural environment of the cell. So these are different categories of live cell imaging assays. You can do a live cell endpoint assay, just one shot, and then you are finished with your result. You can do a cell kinetics. You follow through the experiments over a period of time. You can do a cell fast response assay. You treat it, you incubate, and then get the results. So there are challenges, of course, in kinetic live cell imaging. Every assay has its own advantages and disadvantages. Of course, first of all, it's an expensive toy. Second is how to keep the cell alive for a long time, as I said. So you need to have an incubator attached to your microscope. And of course, there is a problem if you're using fluorescence probe. There's a problem of photobleaching. So in other words, your cells will be bleached. So these are some images in confocal laser microscope. So if you're studying about NATPRAD and its effect on microtubules, NATPRAD and its effect on apoptosis, you can easily obtain beautiful images or NAT products that will affect the membrane of your cells. So these are examples of images before and after the treatment. And from this graph, you can actually compute your EC50 or IC50 very easily because there's a software that will provide that. Another platform now, but still in imaging, is a high-content screening platform. You don't see a microscope here. The microscope is inside this box. So all the objectives are inside this box, and what you see only is a monitor. So yes, very similar to what I mentioned earlier, but this time you don't see the microscope. The microscope is inside the box with all the objectives. And this is usually used for high-throughput researches. So if you want to test the natural product in so many cells, this is the platform available. So indeed, microscopy has affected cell biology and it is able to allow us to visualize the cells and even at the molecular level. And we hope that with advanced microscopy, it will offer us more exciting opportunities to do more research and answer more questions regarding the inner workings of the cell. So this is the theme of the research group, Natural Product Research Group. I also would like to thank the Molecular Cytogenetics Laboratory of St. Luke's Medical Center for allowing me to use the facilities for some of my projects. That's all. Thank you.
Thank you so much, Dr. Enriquez, for excellent, excellent presentation. I'm sure a lot of students are here and they are going to benefit a lot from the wide range of the different aspects or different techniques that we use that you have discussed in your presentation. Before I read out the question to you, I would like to take this opportunity to acknowledge the presence of hundreds of students from India, from Philippines, from other countries who are attending this webinar. As I mentioned to you, you can't see them here as participants because we have a lot of blackout in different parts of Philippines due to repair works. But we have places where the students have assembled and they are watching it on YouTube. Truly speaking, you, the students, you are the honored guests for us, as you all are the most precious assets and treasure of your family, of the institutions, schools, colleges, universities to which you are attached to, and also your country. Your attendance in this symposium during these unprecedented times of global pandemic shows your quest for knowledge and commitment towards finding solutions to problems under all situations and circumstances. May I request you all to send a thumbs up sign or emoticon, as your generation says, on the chat box as a sign of solidarity across borders. We would love to see your thumbs up sign. Back to Dr. Enriquez. Back to Dr. Enriquez. Dr. Enriquez, I have a question from Annie Celine. The question is, what is vital dyes? Can you please explain the importance of vital dyes in your study?
Oh, in our study, we use the resasurin-based dye. And this dye is actually will only color a cell that is alive, that is a viable cell. So once inside the cell is permeable, it can go inside the cell. The cell is alive. There is a reducing environment inside the cell that will change the color of the resasurin dye, a Presto blue, to a, I think I have a slide earlier about blue to red. No? If the cells are dead, they are usually colored blue, and if they're alive, they are in the color pink or red. So it means to say that the cell that is alive is able to reduce the dye, no, from the blue color to the red color. So that's the whole mechanism.